wash, washy, wash, washy, wash, wash your hands. Scrub them while you're singing the song. Wash, washy, wash, washy, wash, wash. Rub your hands and fingers in the places in between. Using soap and water makes your hands so clean. Wash your hands. Make sure you're almost done. 5, 4, 3, 2, 1, now dry.
Wash your hands for 20 seconds, just like Elmo.
Welcome to Chapter 12, Food Safety and Food Technology. Food Technology by Professor Tammy. Learning objectives. Summarize the advantages and disadvantages of producing foods through genetic engineering. Selective breeding. It's like an accelerated selective breeding. It depends what's happening. So if you want to read the article, you can open it up. Here's something about 9,000-year-old breeding that has been done to corn, peaches, and other crops. When you look at it, this is what corn used to look like. And crops that look like before thousands of years of selective breeding. So what watermelon used to look like: corn, carrot, banana, eggplant, and cabbage, kale, broccoli. So when you look at it, food has evolved as well too, based on like, oh, this kind of looks good and tasty. Let's have them breed together and see how we like it. Here's example of what Belgian cattle looks like to increase meat production. This is a normal cow, and this is a Belgian blue cow. Recombinant DNA technology is to obtain the desirable trait, whether through stem cells, clone cells, transgenic organism, sometimes suppressing unwanted traits or splicing genes, or maybe in the future like a Star Trek food replicator. Compare selective breeding and rDNA technology. So selective breeding is when DNA is a strand of genes depicted as a strand of pearls. Traditionally, selective breeding combines many genes from two individuals of the same species. So you get the species with the desired gene you want, and you kind of select these two and say, hey, you breed, and you get a new variety of this new generation of genes, and the desired gene goes into the new generation. Versus rDNA technology. Through rDNA technology, a single gene or several may be transferred to the receiving DNA from the same species or others. So you take the desired gene and you transfer it to the same species, and you get the same species, but with the desired gene that you want. So it looks like species 2, but with the desired gene from species 1 or donor. The promise and problems of rDNA technology. There is the human nutrition of it. There's golden rice, biofortified cassava root. The golden rice gives more nutrients. There's also molecules from microbes and greater crop yields. It can be herbicide resistant or insect resistant when they play with these genes or experiment on these genes. And foods from cloned animals. So if you kind of like the taste of this chicken, you clone it, the clone will taste similar or the same. Issues surrounding GMOs, which stands for genetically modified organisms, are nutrient composition, accidental ingestion of drugs from foods, pesticide residues and resistance, unintended health effects, which is unpredictable, environmental effects, loss of species, because especially if you tend to eat the same foods all the time. Ethics and genetic engineering, and regulations of GMOs. See you soon, and remember to read the book.